Hey gang, back with my last video for this unit, lecture 15, Mitosis 2, starting off on slide 19 if you want to follow along. So the control of the eukaryotic cell cycle. So that whole big cycle, with the G1, S, G2, M, all those, it's a very long, complicated cycle, and it has to be heavily regulated so organisms don't get, messed up. Now, there are three checks. I drew them proportional to where they are in the cycle. The G1 check, which is at the end of G1, the G2 check, which is at the end of G2, and the M check, which is at the end of mitosis. These are regulated by cyclin proteins. Proteins do a lot in our cells, if you haven't noticed yet. So, the evidence for cytoplasmic signals started off with experiments with cultured mammal cells. They use cells at different phases of the cell cycle to form a single cell with two nuclei at different stages. Um, the cytoplasmic signals from one cell would cause the nucleus from the second cell to enter the wrong stage. So that's why there has to be so much regulation, because a signal can mess up like the whole replication cycle. So in this experiment, there was one cell with S in S phase and one in G1 phase. And the result was that the G1 nucleus immediately entered the S phase without going through the G1 check. Um, and the DNA got synthesized. And then in the experiment two, there was an M phase and a G1. Um, and then because of that, the G1 completely skipped the S phase and went right to skipped the S and the G2 phases and went right to mitosis. So it never actually duplicated the chromosome. So the conclusion of this experiment is that molecules present in the cytoplasm control the progression to the S and M phases. Um, so slide 23. Um, so the G1, the checkpoints kind of act as like stoplights at traffic. So without the signal at the G1 checkpoint, the cell enters the G0 phase. Um, and if it passes the G1 checkpoint, it can continue on to the S phase. So the G1 checkpoint is the most important one. If it passes it, it can go throughout the rest of the cycle, and it usually completes the rest of the phases. But if not, it has to switch to the G0 phase, um, which usually just means nothing happens. Um, and it switches to the G0 phase if it doesn't receive that go on signal, I guess. So the G2 checkpoint occurs right before it's about to enter mitosis, and it makes sure that the DNA got replicated properly. So the activity of cyclin proteins is regulated by the other proteins that signal any damage to the DNA. And if there's any damage, then the cyclins get downregulated and it doesn't get to continue the cycle. And then our final checkpoint is the M checkpoint, which occurs towards the end of mitosis. It's also known as the spindle checkpoint, and it makes sure that chromosomes are attached to the spindle complex. Um, it kind of occurs between metaphase and anaphase. So anaphase does not start if any of the kinetochores are unattached. So until all of the kinetochores get attached by the spindle fibers, then anaphase doesn't start. So the attachment of all the kinetochores starts the regulatory complex, and that activates separase, which activates anaphase. It allows the sister chromatids to separate and triggers anaphase. So at the M checkpoint, prometaphase, without the full chromosome attachment, it stops, um, and then it goes on to anaphase, and as long as there's full chromosome attachment, it's allowed to continue on to the G1 phase. Um, Here's a little diagram on slide 27, if you want to draw that. All right, on to control of the cell cycle. So, growth factors influence the cell cycle and they trigger intracellular signaling systems. So, an example would be platelet-derived growth factor. That allows your skin to scab over and heal when you like get a wound, like a cut or a scrape or whatever. It triggers the cells to start dividing to heal that wound. Here's a little diagram. If you'd like to draw it, I highly encourage you to draw any diagrams. They help you out a lot. So here was the experiment that kind of figured this out. So human connective tissue got cut up into small pieces, and it got put into an enzyme mix. The enzymes digested the extracellular matrix, which resulted in a suspension of free fibroblasts. Um, Fibroblasts are uh, something in your body. It looks I forget. I'm sorry. You probably remembered in lecture today, but um, after that, the cells got transferred to the culture vessels. And when the PDGF, which is platelet-derived growth factor, it resulted in cultured fibroblasts. So, uh, control of the cell cycle. So, density-dependent inhibition. That means once the cells reach a certain number, a certain density, number per area, then the crowded cells stop dividing because there's no more room. Most animal cells, that's us, also exhibit anchorage dependence, which means that they have to be attached to something in order to divide. They can't just have like free floating stuff. Um, Cancer cells don't have either of these dependences, and that's what results in cancer is because they're allowed to just continuously divide unchecked, and that's what causes cancer. Uh, so cancer is the failure of the cell cycle control. So when there's no checks during the cell replication, that causes cancer. Uh, there are two types of genes that can disturb the cell cycle that when they get mutated, which can cause cancer. Tumor suppressor genes and proto-oncogenes, like how oncology is cancer research. So, tumor suppressor genes prevent the development of cells containing mutations. So, they keep weird, wacky, messed up cells from actually continuously dividing and stuff because that's usually mutations are almost always negative, detrimental, and our bodies don't like that. Um, so, if uh, any DNA damage is found in a cell, P53 stops that division and nothing happens. P53 is usually absent or damaged in many cancerous cells because it can't like halt the cell division, and so those mutated cells are left to their own devices and can just continuously divide, 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 and that causes cancer cells. There's this little diagram right here. You don't really have to like draw the pictures, but like writing down those little steps is definitely helpful. So proto-oncogenes, some of them encode receptors for growth factors, some of them encode signal transduction proteins. When they get mutated, they become oncogenes, which are literally cancer-causing genes. They're constantly signaled to keep growing and growing and growing. Um, Cancer cells don't respond to signals that normally regulate the cell cycle, like the G1 and G2 checks. Uh, Cancer cells do not need growth factors to grow and divide. They sometimes some of them just make their own growth factor, and some of them just pass the growth factor signal without the presence of the growth factor. It's a very abnormal cell cycle about because it just like keeps going and going and going. And then our last slide here, transformation, is the conversion of a regular cell to a cancerous cell. If it can avoid immune detection and forms tumors, uh, which are the big, masses of weird abnormal cells that are inside normal tissue. Like how if you get a tumor in your stomach or your prostate or your brain, it looks it just like a giant mass coming out of your whatever organ it is. Benign tumors um, are if the tumors only remain at the original site, so if it's localized to that organ, whereas malignant tumors start spreading around the whole of your body. They invade the surrounding tissues and undergo metastasis, which can form additional tumors. Not fun. Cancer sucks. Uh, that is going to be it for Mitosis 2. I wish you all good luck on your exam. You're going to rock it. Uh, I'll see you guys whenever.